Yeah.
She said that's recorded.
Well, oh. Then I don't think mine recorded.
Good afternoon. I had to make sure Brandon couldn't leave. It's my pleasure. It's my pleasure to introduce you. Schumacher Clark. They're a new law firm that does DNA annotation. No, I've been. These two are in very different circumstances. Daniel has already finished and is heading to medical school in the fall. This is one of his last requirements, so we appreciate that before he walks in just a couple weeks. And he started this project really this semester. Camille is going to finish up in the fall and so is a sort of half senior along the way. But I just appreciate what's been wonderful about them on this project is they are the first people initiating this task. They were given very little supervision and lots of encouragement to explore. So what they have learned in this project is really their discoveries. For some of them, I mean, I may be the advisor and mentor on this, but I got to learn after they discovered it. That's when I could find out the answer. So I've just appreciated, they've just done a really fine job on doing this, and we're looking forward to both of them being the kind of doctors that we would all like to have because they can do this kind of discovery, independence, and they care and are dedicated. So I've just been very grateful for you. You're great students, and I hope you can enjoy their talk.
We also hail from Colorado.
And they hail from Colorado.
Thank you, Dr. Chase, and we'd like to thank all of you for attending. We'll be putting your paychecks in the mail later this week. Thank you for the pity laugh. So, just wanted to start us off. It seems like there's an increasing trend, or at least a steady trend, of seeing newspaper clippings and headlines and stuff like this in the news, where we have meat corporations apologizing for recalls and individuals being affected, and E. coli crises and outbreaks. And so for me, my main role in this is as I'm a consumer, and I don't want to be the one finding out that a new pathogen has evolved. And so it affects us as consumers. But if we take a step back and look at it at an agricultural level, the way it impacts agricultural communities such as Idaho, and the U.S. as a whole, the implications of pathogenic strains of E. coli are more than just for like myself individually. So with the dangers of this pathogenicity and of new strains evolving, early identification of new pathogens is key. And that leads us to U of I found this mystery strain. We deemed it a mystery strain for dramatic suspense purposes. It's really just another strain of E. coli, but we named it mystery because it sounds cooler. It's a strain that they found in the recto-anal junction of cows, healthy cattle. And we don't want to say recto-anal junction the rest of the day, so we're changing that to RAJ1. We figured you want to keep your lunch down as opposed to throwing it back up. So this strain was taken from cows, and it was found to produce a Shia toxin, which is one of the virulence factors we'll discuss later of pathogenic E. coli. But it was not, when it was sequenced and further looked at, it was not found to be a strain that was already sequenced and known. So this raised a bunch of questions, mainly how close is this strain to evolving to become a pathogen and causing another outbreak?
Okay, so before we can fully understand the whole purpose of RAJ and its pathogenicity as compared to other dangerous E. coli strains, there are two things that we have to study prior. And the first is genome annotation, which I'll be talking about, and the second is E. coli pathogenesis, which Dan will be talking about later. So, first things first, what is genome annotation? Essentially, genome annotation is the retrieval of a DNA sequence to identify and name the genes that are involved, and that first starts with genome assembly. So genome assembly is going to take short fragmented sequences of DNA and connect them into contigs. And essentially, I mean, you can see from the cute picture, oh, no, I clicked the wrong thing. But there was a cute picture up there. Yeah, there it is. This picture just essentially shows a bunch of character fragments that are put together to create one long consensus region of a sequence. And for the RAJ1, what we did was we took the Roche software and assembled the RAJ into about 387 contigs. And again, like I said, these are broken up pieces of sequence. So when you look at this total picture, this is the laser, that's what I was looking for. You can see these sequences and they go together and they fit like a puzzle piece, but we haven't yet determined what the total puzzle piece looks like. And later on, Dan will talk about it in the E. coli pathogenesis section, but what we're comparing RAJ2, which is the Sakai, has not only undergone the sequence contig, but has also been scaffolded and basically created into an entire genome map. So all of its pieces are put together into one solid contig, whereas our RAJ sequence at this time is still in the contig stage. So after you have assembled your genome, the next thing you want to do is annotate it, and there are various steps that are involved. So for RAJ, we used the RAS server, and we went through these steps. So we loaded the sequence into the server, we created a database through it, and RAS began to predict the genes and then blast it, which basically means just find the genes and their functionality. So once we've done all of those things and we have all of our genes together, we were able to use the seed viewer, which is just kind of a subsystem of RAS, to compare it to various species. And that's very important to what we're doing here. So I've taken genetics lab with some of you, and so we all know that when we took genetics lab, we focused on one specific gene. We studied each gene's functionality separately, individually, of that one single genome. However, what the seed viewer does is not only lets you look at the functionality of an entire genome, but lets you compare it with other systems. So not only could we look at RAJ, but we can also compare it to the Sakai. And so that becomes very important for what we want to do here. And now it's Spencer.
So now that we kind of discussed genome annotation and what that entails, the other part is E. coli pathogenesis, and then where these two meet is sort of where our project headed off in its own direction. So we all learn in science classes and stuff that E. coli kind of has two forms. That's a bad way to describe it. But there's good E. coli and there's bad E. coli. We all have E. coli in our bodies. Animals have it. It helps us with digestion and normal healthy functioning. But when it becomes pathogenic, that's when the negative side effects occur. So the specific pathogenicity that we're looking at is called enterohemorrhagic E. coli. And the way this works is when we ingest undercooked or raw meat, we house it in a zoologic grow and embed in our digestive epithelium. And from there, it releases its virulence factors and toxins and whatnot, causing things ranging from abdominal cramps, muscle spasms, all the way to diarrhea, fever, vomiting. And in some cases, it has been noted to go all the way through to hemolytic uremic syndrome, which includes renal failure and can eventually lead to death in about 3 to 5% of cases. So it's kind of important to understand why we're studying this. So like we said, E. coli 0157H7 Sakai is the genome that we know is pathogenic, and so we're kind of comparing the mystery strain to Sakai. This is the most common and most known enterohemorrhagic strain in North America, and this is thought to have evolved from gene transfer from another pathogenic strain of E. coli. And that's important because E. coli is able to transfer genes between different strains, and that's how a strain like RAJ1 could go from being normal E. coli, good E. coli, to pathogenic bad E. coli. So we took RAJ1 and Sakai and compared these at a nucleotide level, a genome level, and what we did was we aligned the RAJ1 strain with the Sakai strain, and that's what this graph shows. And as you can see, mostly we see blue, which when you look down here is about 80 or 98 to 100% identity match. So our motto with this was the bluer the better, and what we see, the majority of this is blue. So what that says, we estimate that 95%, we have a 95%, average 95% sequence similarity. An interesting thing to note is the difference in size of the genome. RAJ1 is about 300,000 base pairs shorter, which we're, first, like that's a lot of base pairs to be missing. But as of now, it seems that all of the coding sequence and pathogenic factors are coded for in RAJ1, despite its missing sequences.
So not only did we decide to look and compare RAJ1 and Sakai at a genomic level, but we also decided to look at it at an organismal level. So we used a phylogenetic tree to do that. And essentially, for those of you who don't know, a phylogenetic tree is going to act as a bloodline, sort of. So you're going to see relationships between the query or the organism that you're studying in relation to other, in our case, bacteria that have undergone lateral gene transfer to create new phenotypic species. So when we look at the Sakai, we see that its closest relative is the Klebsiella pneumoniae, and then it's also related to various Serratia and Yersinia bacteria. And then what was very interesting and promising to us, because we were looking for similarity here, is that our RAJ1 phylogenetic tree also showed Klebsiella as being the closest relative, also showed various Serratia and Yersinia bacteria, and then there was also the Enterobacteria, which hadn't shown up in the Sakai, but it occurred so few times that we figured that it was somewhat insignificant to what we were doing. So, looking at these two strains, you can clearly see that we've just made a color change. Blue is Dan's favorite color, so we had to make it blue. But all of this is to basically say that Sakai and RAJ at this surface level look exactly the same. They appear to both make Shiga toxin. They are related to the same things. They have similar genome sequences. But what's very important, and the most important part of our research, is does it have these virulence factors that we're looking for? And as I said, we already see that Shiga toxin is there, but we know nothing about the other 5 that are there. So what Dan and I decided to do was choose one of these factors to study and further understand the similarities between RAJ1 and Sakai, and we chose hyperadherence genes.
So what we discovered with this virulence factor is this is the way that interrohemorrhagic E. coli is able to attach to the small digestive epithelium, and that's where its pathogenicity comes from, like I said. So it's kind of the functionality part of how this works. And what we discovered upon further looking is that it's mediated by a hyperadherence cluster. So this cluster consists of these 7 proteins. And this is taken from Sakai, where all 7 proteins are sequenced and known. But if we use the CPUR, like it's a way of comparing genomes, and we look at RAJ1 compared with Sakai, these 6 were found with known locations on the RAJ1 genome, whereas the question mark on YID-E was kind of what drew our attention to focus on YID-E. So we looked further, found the sequence, the amino acid sequence for YID-E, and kind of went from there with our research. That's the reason.
So, as we said, the CPUR gave us this amino acid sequence. And what's important to note, because I know that the question mark for a while kind of stumped me, is that there was an amino acid sequence given for RAJ1. There was just no official known location of where it was. And what's important to note about this is that the YID-E in RAJ1 is 98% identical to Sakai's. So there were 553 amino acids in the sequence, and of those 545 were exactly the same. However, there were additions and truncations that were found on RAJ1 that kind of made us suspicious and skeptical as to if we could really determine if this was YID-E. So the RAJ1 has this addition of these 8 amino acids up here and is missing these 8 up here. And taking biochemistry, we've had a really great opportunity to fully understand amino acids and to bring that knowledge in, so that was kind of exciting. But we looked at this missing portion first, and we noticed that there are a lot of non-polar hydrophobic amino acids there. There are a couple that are polar in charge, but mostly we see valine, leucine, isoleucine, glycine. Whereas when you look at this top strain here, we see a lot of polar charged amino acids. And so looking at that made us very suspicious and led us to these 3 major questions. The first question is, according to the amino acid sequence that was given, do we have YID-E or do we have a completely different protein? The second question was, do those amino acids change the folding 3D structure of whatever that protein is? And finally, is that protein found in a different place than the YID-E of the Sakai? So what we were able to do was use IMG ACT Toolkit because it gives us web-based tools that help us to further identify this strain in comparison to the Sakai. So we looked specifically at protein blasting for sequence-based similarity, which was going to show us the best protein alignment that made sense for what our unknown was. And then we also used a transmembrane helical Markov model, which just sounds like a big blur of words, and Phobius. And both of those essentially helped us to figure out the folding structure of the protein as well as where it was located. So just to take a step back and kind of remember where we've been and where we're going, we have just understood genome annotation and E. coli pathogenesis to understand RAJ1. We're going to take that information using the toolkit to further compare YID-E between RAJ1 and the Sakai. So when we did the sequence-based similarity with the BLAST search, we found really great results. In fact, it showed that out of the thousands of genomes that or the thousands of proteins that it could possibly be, that RAJ1 was closest with YID-E and it had that hyperadherence deviation. And so that was very important to us because it really answered our question of, is this really YID of everything that we could have possibly found? And so that was really positive for our results.
The next question we were looking at was 3D structure of the protein, because we all know that if structures, or if proteins don't fold right, they don't function right. So this model allowed us to input the sequence and then get a graphical representation of the folding patterns and transmembrane helice section. And these graphs look nearly identical, but there's a little minor differences which show that they're actually different. But it's really positive to see almost identical graphs, meaning that these proteins do fold in a similar, almost identical way, leading us to assume that functionality would also be the same. The phobias is similar. It allows us to compare folding in transmembrane helices as well as signal peptides. And this we thought was going to be interesting because signal peptides are usually coded for towards the beginning of a sequence, and since we saw that addition and truncation, we were wondering how that would affect this in particular. But again, we see almost identical peaks. This red line down here demonstrates the probability of there being a signal peptide. And so in RAJ1, there's absolutely none, it's 0. So there's no signal peptide there. But the one difference we see is Sakai has a peak right at the beginning of around 0.25, give or take. So we thought maybe that's interesting, but when we read up more about the background of the phobias test, we were told to throw out any values less than 0.7. So this is clearly well below 0.7, so we think this is insignificant, and there's not a signal peptide that would cause different function in Sakai versus RAJ1. So in conclusion, we think that RAJ1 does have YID, and this comes from the test we ran to answer those 3 questions. The first question being, is it actually the same protein, or is it something, is there a better match, which we proved it seems to be YID. Both proteins fold in a similar manner, and they both are found in the same location on the cell, which is transmembrane. So, in light of all the data that we've collected, we're fairly confident in saying that this protein sequence does code for YID. And since RAJ1 has it, it has an extremely high potential of becoming or being a RAJ1 reactor. So back to the beginning, well, not really, the middle. The only thing we knew that RAJ1 had was this Shiga toxin, and these are the 6 virulence factors needed to become pathogenic. So now, based on the tests we've run and the data we've gathered, we're confident in saying that hyperadherence is coded for within RAJ1, and so we're checking that one off the right list.
So the further questions and moving forward, Dr. Chase does have more plans to continue to study RAJ1 and not just YID at this point, but the other virulence factors that were involved. So something that we think is also very important is it's really great to have the computer models to help compare and contrast Sakai to RAJ1. But what's really important after those steps is to be able to do wet tests and introduce it into a lab to really understand the enterohemorrhagic properties of RAJ1 if we do realize that they are really similar. And so finally, we'd like to do our acknowledgements. We'd like to thank all of these sites that helped let us use their products. We'd like to thank Drs. Minnick and Bohosh, who gave us the strain note from U of I. We'd like to thank Dr. Chase because she helped us dramatically with our PowerPoint. It wouldn't be nearly as great without her. We'd also like to thank Project Free TV because it kept us up many nights giving us Disney movies to watch while we ran all of these sequences. And so, unless you have a really hard question, we'd like to be taking questions at this point.
If you have a PhD, you're not allowed to
Yeah,
ask questions.
hands down.
That's wrong.
You don't have to listen to them.
I was just kidding, that was just funny.
Unless we understand, then you can ask questions. Yes, Dr. Miguel.
They took my PhD away, so
Oh, very good.
Well, there you
I
go.
moved over there.
Ask
All
away.
So it's a collaboration with the University of Idaho. I know that that's some of the research that they do there. How does this fit into their bigger picture research that maybe that your part here can move into a communication of some type, publication? Because this looks like it seems that the word of this discovery needs to get out in some way.
So we mentioned a little bit the further research, because we found we're confident saying YID-E, one protein coding for hybrid adherence, is found in RAJ1. So we would need to go through those other four, which is the virotoxin, flagellar motility, and the other two, I forget which ones those are. But those four would need to be like similarly tested, which is the future research.
So you have to do all four, you think, before you can publish. That's true.
I would say yes, because those are the major properties that cause the enterohemorrhagicity. So without those, I mean, it's really great that we have YID-E, and that's a very important piece, but I think that altogether that would really cause, or give a cause for communication between you and a need for that. Dr. Schumacher, if you do have a question, you can ask it. We were just